And so Jasper Therapeutics was founded by Dr. Judy Chajuru and Dr. Susan Prohaska from Stanford. And I think Dr. Prohaska is on the line today, so I think she's waving. And unfortunately, Dr. Chajuru, because of a last-minute family issue, couldn't join us today. But she is with us today in her thoughts, and she'll be available. If any of you have questions, feel free to reach out to her at any time. But they founded this company based on the principle of making stem cell transplant, essentially, like I said, better by making it safer. And when they took this program out of Stanford and started Jasper, they recruited a large team of very experienced professionals to be able to launch this program into clinical trials and to get this therapy into patients. So I think here I'd just like to take this opportunity to introduce some of the members of the team. Dr. Kevin Heller, who's online, I think he's waving as well, is our Executive Vice President of Research and Development. He's a pediatric hematologist oncologist by training, and he's very involved in terms of the clinical development and bringing this drug to more and more patients. Kevin, would you like to say a few words?
Yes, now that I'm allowed to be unmuted, yeah, I don't want to hold up the presentation, but just happy to say that I'm very excited to be a part of the Jasper team and have the opportunity to introduce this exciting program, because it is so important for us to make an impact on patients with SCID. And as a pediatric hematology oncologist, it's hard to believe I'm this old, but many years ago when I was younger, I saw the value of transplant, but the incredible risks that are associated with it. And this is a great opportunity where we may actually be able to shift that the risks associated with transplant and actually get that healthy transplant available to more patients, especially a lot of the children who are starting out life with SCID. So.
Thanks, Kevin. And then I'd also like to introduce a couple of other members of our team who are here today, if they could just wave. We have Janet Hurt, who's our Vice President of Clinical Operations. On her team is Nicole Harada, who is the clinical trial lead for the SCID trial. And I introduced to you Dr. Susan Prohaska, who's one of our co-founders and a senior scientific consultant for all of our scientific endeavors and projects. And then I think many of you might meet other members of our team as we go forward. So just to start, severe combined immune deficiency, I'm sure all of you are familiar with. It is an issue of the blood-forming stem cells at heart, because the blood-forming stem cells, they're also called hematopoietic stem cells, and sometimes you'll see on our slides they're termed HSCs. These blood-forming stem cells, they live inside the bone marrow, and they're responsible in every one of us to make all of the blood cells for all of our life. And so they make red blood cells, they make immune cells such as lymphocytes, the T and the B cells, and these immune cells are required to be able to help us fight infections. And in patients with severe combined immunodeficiency, they can't make the normal lymphocytes, the normal immune cells, T and B cells. And so these patients are at risk of developing infections and being unable to deal with many kinds of infections. And transplant is a procedure that replaces the hematopoietic stem cell or the blood-forming stem cell in SCID patients who don't make normal lymphocytes, replace them with healthy stem cells that are capable of making these T cells and B cells that can fight infection. So the process of blood stem cell transplantation really requires a couple of steps. It's not a one-step process. And hematopoietic stem cell transplantation can occur in both what we call an allogeneic setting or an autologous setting. All that means is, where do those stem cells come from? So those healthy stem cells could come from somebody else, like a brother or a sister or a parent or someone unrelated completely, and that would be allogeneic. Or they could be autologous stem cells, which means they come from actually the patient, him or herself, but they undergo gene modification to correct them. And sometimes this is just grouped under the bucket term of gene therapy. And so we have a source of healthy stem cells, but before you can give them to the patient needs to undergo a process that's called conditioning, which is preparation of that patient's body, of the bone marrow, to receive the new donor cells. And the reason we need this preparation is there's evidence to suggest that you have to essentially clear up the space in the bone marrow so that there's space for those new cells to come into. And if that space is occupied with the recipient's stem cells already, then the donor stem cells don't have any room to come into. And so that's when they don't engraft, right? Those donor stem cells, those healthy stem cells that we want to get into the patient to provide the cure, basically, they're not able to because they can't find homes. The homes are already occupied with the abnormal or the defective stem cells. And so these patients get a process called conditioning. And the conditioning usually involves some form of chemotherapy or radiation. And it depends on the recipient and the condition of that recipient, what exactly would be given. But if we can get that healthy donor stem cell in, then we could potentially cure a number of diseases, including severe combined immune deficiency. And so Jasper is really focused on trying to make this conditioning piece safer. Because right now, the chemotherapy, and the most common one used in severe combined immune deficiency patients, is called busulfan. And sometimes radiation could be used in certain groups of patients too. But these agents, as a whole, they do make space in the bone marrow. So you get the space opened up for those donor cells to come in. But unfortunately, they also cause a lot of damage to cells not in the bone marrow. And it's this cell damage that causes tissue injury, like damage to the liver or to the gut or to the kidneys. And it's this damage that does lead to an increased risk of a cancer developing later in life. So Dr. Shadru at Stanford pioneered the development of an antibody therapy. And this antibody therapy is named JSP-191. And this antibody selectively targets the blood-forming stem cells and leads to those stem cells to become depleted. And we get that empty bone marrow space that we want. So space for new healthy stem cells to come in. But the good thing about this antibody is that it doesn't cause what I'm going to call collateral damage to all of the patient's other cells, their kidney cells, their liver cells, anything else. So the toxicity is much lower compared to the chemotherapy or the radiation that might be used traditionally to prepare a patient for a blood stem cell transplant. And so, just to make sure, we all get on the same page about why this antibody really works, is that the blood-forming stem cells, they have something on their cell surface that's called c-KIT, or CD117. And it's basically a way for the stem cell to say, hey, I should still be a stem cell. I should stay alive and make blood cells and make immune cells. And they get this signal from their environment through a molecule called stem cell factor, or SCF. And so this antibody, JSP191, it blocks this survival signal for the stem cells. And so the stem cells get depleted when you give this antibody. And the empty homes, in this case, it's depicted as an empty bone marrow niche, but the empty homes are open for occupation by the incoming donor stem cells that are transplanted in. And so the hypothesis that Dr. Shizuru took to the clinic was to use this antibody to prepare severe combined immune deficiency patients for transplant. And this is the clinical trial that was started at Stanford by Dr. Shizuru, looking at this antibody as conditioning or as preparation for the bone marrow for transplant of SCID patients. And we've studied so far 2 separate SCID groups. We call cohort A, essentially, the patients who actually had a transplant as a young child, but they didn't recover their full immune cell function. So they were still susceptible to infections. They still required intravenous immunoglobulin. They still had symptoms and signs of SCID. And so these patients were enrolled on the trial to basically give them another transplant, a retransplant. And this time we used the antibody as the conditioning before we gave the donor cells. In a separate group of patients that we just recently started studying are the newly diagnosed SCID patients. These patients have been identified through newborn screening, which is an amazing program. I think that Heather has played a critical role in promoting and launching in all 50 states in the United States. So we are also enrolling in a separate cohort, cohort B here, the newly diagnosed severe combined immune deficiency patients. So these patients haven't had a first transplant. So here in this trial, we're really looking for the ability of our antibody-based preparation of the bone marrow to allow the donor cells to get into the bone marrow. And we measure that by what we call chimerism. And that's basically the percentage of the cells that we find in the blood that come from the donor. So these would be the healthy stem cells. And we also look for evidence of normal immune cell function, normal B cell function, normal T cell function, that would suggest to us that we are making a real impact on these patients' lives in order for them to be able to control infections and respond to infections appropriately. So to date, we've actually dosed 12 patients in the retransplant setting. So all of these patients had a first transplant, but after that first transplant, they still required intravenous immunoglobulin. A number of them still had issues with chronic infections. And so these are the genotypes that we've treated so far. You'll see that there were a couple of patients who had Artemis SCID, a couple with XSCID, a couple with RAG1 deficiency, and another patient with a PRKDC mutation. And their ages really ranged. The youngest patient in the retransplant group was 3 years old when she received the transplant with JSP191. And the oldest patient we've dosed to date is 38. They had a variety of donors, and these donors were the same donors as their first transplant. And now we've had follow-up in some of these patients for 3 years. And I'm really excited to be able to show you the impact we've been able to have on some of these patients. So first of all, I think it's really important to point out that all of these patients have done really well during the transplant. There have been no related serious adverse events to date, and there have been no infusion reactions to the antibody. And even though the first patients on this trial, because this was a brand new drug in a brand new patient population, they were dosed in the hospital, so they had to be admitted to the hospital, and then they were followed in the hospital for several weeks, actually, just to make sure everything was going okay. Those first patients that we admitted, their biggest complaint was boredom. So we ended up actually amending the trial so that these patients now come into the hospital to get the antibody, and then we monitor them for the first 2 days. And then they can actually be discharged, so they don't have to spend the night in the hospital room. And then they come back into the inpatient or in the hospital setting when it's time for their donor cells to be infused. And so, like I mentioned, we've dosed 12 of these retransplant patients to date. And these are the patients that we have the longest follow-up on. And you'll see, among the 6 patients that we have really long follow-up on, 5 of them actually have, new, what we call naive T cells. And these are important because these are T cells that can respond to new infections, new pathogens, and new bacteria and viruses that emerge in the community. Things like, I would say, SARS-CoV-2, for example. And actually, 2 of these patients, they don't require intravenous immunoglobulin anymore. And they've actually been vaccinated with some of the traditional childhood vaccines. They were vaccinated, in this case, in their teens and in their 20s with vaccines, I think, probably some of us received as babies. But it's great they actually responded to the vaccines, and that indicates to us that they have evidence of normal immune cell function. They're doing what their immune system's supposed to do. A number of these patients, their chronic infections have resolved. Chronic diarrhea is resolved. Chronic sinusitis, upper respiratory tract infection has resolved. Patients are gaining weight and doing overall, I'd say, very well. And so we're very encouraged and cautiously optimistic for all of these patients. This is data for actually one of our newly diagnosed SCID patients that we treated on our trial recently. He was an ex-SCID child. And now we're very excited because this patient now has evidence of blood stem cell engraftment. So this is how we measure blood stem cell engraftment. We measure basically in their blood, what fraction of some of their cells are coming from the donor. And so this is very exciting for us because we know that now this patient has donor cells in their bone marrow, and the donor cells in the bone marrow are making normal immune cells. And here, this SCID patient actually now has normal levels of T cells for his age. And the T cells actually also function. So in one of the laboratory studies that's very traditional for the diagnosis of SCID, it's called a T cell function test. And one of the criteria to diagnose SCID is that the T cell function is low or abnormal. And this patient's T cell function is normal now. So we're also very encouraged and very excited for this patient. So just in conclusion, what we've seen in the, on the science side as well as on the clinical side is that we can use the targeting of CD117, this receptor, this signaling pathway on blood stem cells, and use an antibody that can target that stem cell, target that pathway to basically replace the need for chemotherapy or radiation in stem cell transplant. And this way we think we can significantly reduce the toxicity while maintaining the efficacy or the potential curative potential, of blood stem cell transplant. And so our early data from this clinical trial really, I think, has shown, that this agent, this JSP-191, is well tolerated, and we can get stem cell engraftment. And we're seeing, evidence of clinical benefit in these patients, and we're very excited about that. And this trial is open to enrollment for newly diagnosed patients as well as retransplant patients. Here are some of our clinical trial site principal investigators at their various institutions. Stanford, at UCSF, Memorial Sloan Kettering, University of Minnesota, Children's Hospital of LA, UCLA, Cincinnati Children's, at the National Institutes of Health, and at Children's Healthcare of Atlanta. And then just to provide you, I think this is the specific contact information. If you or your family or anybody that you know might be interested in learning more, please feel free to contact these sites and these principal investigators. So I think with that, I'd love to pass it on just really briefly to my colleagues Janet and Nicole, who can tell us, just briefly about some of the programs that we have to support patients who enroll in our study.
Thanks, Wendy. Heather, did you want me to go ahead and just sort of briefly talk about the travel guideline, or do you want to field questions first, or how?
I've got lots of questions, but I want to make sure that they hear about the travel guidelines too. So let's go ahead and proceed with that, if that's okay.
Absolutely. So really quickly, any patient who qualifies for the clinical trial, Jasper does have in place pretty extensive travel guidelines. It outlines, basically it covers reimbursement for travel related expenses for your study visits for the actual patient, and if the patient is a minor, of course, one additional parent or guardian to accompany each child to their required visits. The way that reimbursement works is through the clinical sites. So Jasper cannot reimburse any patient or family directly. So all of that happens through each of our clinical sites, and they have a system set up in place where you submit your receipts and documentation of any of your expenses, and that gets paid through our partner, Precision for Medicine, and Jasper then reimburses them that way.
And that's huge for those of us that, have had to do air travel, maybe to get to a clinical site. So that's something to consider. And I know that maybe air travel is a little bit scary right now with COVID, but hopefully with more and more people getting vaccinated, that won't be, that will end up becoming a thing of the past. But I think that that's a great reimbursement strategy, and it sounds very similar or parallel to sort of what the NIH has offered. So those of you that have been to NIH before maybe have seen a similar travel plan like that before. So that's nice. That's really nice. So thank you for sharing that. Anything else on the Jasper side, or shall we go to questions?
Well, yeah, let's go to questions.
Let's go to questions. Okay. So I'm going to, can you stop sharing your screen?
Of course, yep, stop, share.
Okay. And I'm going to put up mine. Whoop. There we go. We don't want to see welcome again. I want to thank Jasper Therapeutics for taking time out of their busy schedule to prepare this excellent presentation and share the results with us here this evening. My hope is that all of you will leave this evening with an understanding of who Jasper Therapeutics is, what JSP 191 is, what its role is as a conditioner, where the trial sites are, and that the trial is currently open for enrollment. Now we're going to start with the Q&A session. I've got lots of questions. We'll go with the first one here, and the first one is, how are the JSP 191 antibodies made, and are they grown or manufactured?
So the antibody is made in a very, I would say, traditional antibody way, and they are grown in a cell line by one of our collaborating organizations. They're called Lonza. They're a big company based out of Europe.
Okay. Another question. How long are the JSP 191 antibodies active or alive? And then the other question is, and then after the infusion, how long do they live in the body after the infusion?
Yeah, it's a very good question. So the JSP 191 antibody works almost immediately when it goes into the body by blocking that critical survival and maintenance signal for the blood stem cells. And we wait a certain period of time. And for example, at the 0.6 milligrams per kilogram dose, which is the current dose cohort that's open, we wait approximately 9 to 12 days before we give the donor cells. And we actually determine the exact date by basically collecting blood samples immediately after the antibody infusion. So we can measure exactly how much is left in the body at a particular time and model out when the antibody gets to a specific level where it's safe to infuse the donor cells. And by the time we infuse the donor cells, the antibodies are not active and they don't interfere with the donor cells.
Okay.
Hey, Heather, may I add something? Like, because the person who asked the question asked, how long are they alive? So the antibodies are not really living. So, I think that's probably a part of also the reason that we've been fortunate enough that we don't see any side effect profiles. The antibodies are not, it's not cellular therapy. It's not like a vaccine. And as Wendy described, thankfully, we have not seen any significant side effects associated with the single dose in the conditioning setting.
Great. Thank you for clarifying that, Kevin. All right, the next question is going to reference slide 9, so that's why I wanted to bring this up again. Can you explain in more detail why reticular dysgenesis, ADA, and PNP SCID are excluded from both cohorts of the trial?
Yeah, no, it's a very good question. I think, when the trial was initially designed, we had to evaluate, which patients would be most appropriate for this kind of conditioning. And if you recall from my other my other slide, the mechanism of action of this antibody is really directed at the blood-forming stem cells. And so patients that have a risk of rejection because they have some low-level immune cell function, they were not initially included because using our antibody as a single agent we felt might not be strong enough to help get those donor cells in. So we were concerned about rejection of the donor cells using our antibody conditioning alone. That doesn't mean that going forward, we are actively looking at ways to combine the antibody with something low dose. It may not, we are not looking to combine it with, say, busulfan or anything like that, but something else to enhance the ability for the donor cells to engraft so we can overcome that potential for rejection. And we're actively investigating this and seeing what partners would be good for JSP-191 conditioning. So it's still safe, very low toxicity, but then we're not faced with this potential for graft rejection.
Great answer. Okay, this is also, I understand inadequate B-cell engraftment would include those still on IVIG, but what do you consider incomplete T-cell reconstitution?
Yeah, that's a very good question. So I think in a number of these cases, sometimes we do see some T cells in patients who received a first transplant, and they tend to be what we call memory T cells. And that's basically the immune cells that are long-lived, but they can only address very specific infections that they've been selected to address. There's no new immune function to deal with new viruses, new bacteria that someone might face. And so that's what we call incomplete T-cell reconstitution, because sometimes these patients, they do get some memory cells, but we don't consider that real full immune function because those memory cells can only address a very limited repertoire, limited types of viruses or bacteria or anything like that.
Okay. On this slide, what do the two green lines on slide 12 represent? Are they the SCID patients, and one looks higher than the orange one, and how are they clinically? So that's like 4 questions in one.
Yeah, so That's a very good question. So yes, actually, the two green ones are the SCID patients.
Okay.
So one of the SCID patients, he received the lowest dose. So that was our starting dose in the trial, 0.1 milligrams per kilogram. Even though he actually didn't have a significant, a robust naive CD4 T cell development, his chronic norovirus diarrhea has improved, and he's also gained some weight. But I would say, the response, he's still on IVIG, and, he still required a course of antibiotics. Whereas, the other green SCID patient is, doing, I would say, a little bit better, relatively speaking. I mean, each of these patients come into the trial with their own sets of, different types of chronic infections. But that SCID patient is also still on IVIG, but the clinical status in terms of the chronic infections appears to be a little bit better, at least anecdotally, and we're just continuing to follow that patient.
Okay. So on the same slide, in the orange box and the red box here on the right, you mentioned a little bit that they respond. The responses to vaccines that you mentioned on slide 12, were those live vaccines, and if so, which ones? I know you said something about the ones that we usually get in childhood, but were they live or were they not?
So we started with the ones that were not live. So they actually both were vaccinated against tetanus, Hib, so Haemophilus influenzae B, and they both developed responses to those vaccines. And the plan is to continue, vaccinations over time and just see how they do.
Okay. All right. So none of the patients have gotten live vaccines then yet, correct?
No, not yet.
Correct. Okay. This slide, on slide 13, regarding the newly diagnosed patient, to what do you attribute the more robust response in comparison to the retransplanted patients?
Yeah, so I think, it's partly to do with that these patients are infants. So this infant was actually transplanted when he was 6 months old. And in our retransplant patients, they ranged from 3 years to 38 years. And in infants and in very young children, the immune organs, called the thymus, for example, they are much more robust. In people such as myself who are much older, the thymus is essentially retired. And so it's not so active in terms of making all of these new T cells. And it's really the thymus's job to pump out and make all of these new T cells. So we think in older children and in adults, the T cell recovery is going to take longer. And that's why in slide 12, we've had to follow these patients, some of these patients, for years. And in fact, the patient who was transplanted in his 20s, it did take about a year and a half before we started seeing T cells, but they did start coming in. And I think that's partly because we had to wait for the thymus to come out of retirement, so to speak.
Okay. How many years will you follow these patients to gather long-term follow-up data on?
Yeah, right now in the trial, we are following these patients for 5 years post-transplant. We do follow much closer intervals in the first 2 years, and then after that, it's 6 months and then every year. So the follow-up is spaced out more after 2 years.
Does your travel policy plan, does that go for follow-up visits also?
Yes, definitely does.
Okay. How do the patients feel after administration of JSP 191, and what does well tolerated mean in real world terms?
Yeah. So they've gone through the infusion without any problems. It was actually smoother than a blood transfusion. It is through the IVs, and so through the vein. So they do get a catheter inserted, and then that's where the cells that will eventually also go through. So by well tolerated, we mean that they haven't had any reactions. They haven't had changes in, significant changes in what we call their vital signs. So their, blood pressure or anything like that. There's been no allergic reactions to the infusion at all. So that's what we mean by well tolerated. And then after they get the antibody, the first couple of patients, they actually had to stay in the hospital because that's what we required, because we wanted to monitor them very closely. And they were just hanging out in the hospital, and the nurses would come up to us and say, "So why are these patients here? What are we doing for them?" Really nothing. They were quite bored, actually. It was one of the complaints we got. "Can you please go home?"
So, Wendy, would you want to compare it to like the IVIG infusions? Because a lot of these kids do get that infusion. Wouldn't you say that it's somewhat similar as far as the process, and this is something that they are familiar with, and they shouldn't expect?
Yeah, very much so. Yeah, that's a very
so like that's an upside, and I like what Wendy said, that the usually with some of the other agents that are used for conditioning before a transplant, the kids or the patients go through quite a bit of discomfort that require them to be in the hospital. So we were happy to say that boredom was the side effect, and that thankfully it went very smoothly, and it continues to be that way. So
great. I'm not sure what slide this was on, but they said in the line chart on the previous slide, it showed JSP 191 given at day -10. Is it a 1 day, 1 dose, or was it given several times before day 0?
Yeah, it's actually a one-time dose, and it lasts about 1 hour. And it is given, so that picture really shows what happens if you are given the 0.6 milligrams per kilogram dose. And it's essentially about somewhere between 9 to 12 days, usually around 10 days, until you get the donor cell infusion. And you're not stuck in the hospital anymore during that period. The first couple of patients they were, but now, you can go home after 2 days, and then you can come back for the donor cell infusion.
I think that was on slide 8, right? So
okay.
That's yeah, it does it. Oops, sorry, I guess it was slide 9 because we inserted an extra slide.
Yeah, this one. Okay. But by going home, you mean if you've traveled, you mean some place close by till you can actually go home, correct?
Yes, that's right. Sorry if that was confusing. Yes, so you can go to the local housing if you're traveling in, and be with your family. You're not hooked up to any machines or anything.
The pandemic's over. Go out to a movie, maybe.
Yeah.
not be stuck in the hospital. That's a nice bonus if you're not stuck in a hospital.
Why are original donors required, and do you in the first cohort, and do you intend to advance trials to those without original donors?
Yes, so the original donors are required right now because our antibody doesn't address any kind of immune rejection. Basically, if a patient has a response to a different donor, then we could see failure to get those donor cells in. And we do have an intention of exploring ways to transplant patients who don't have an original donor, and that's along pretty much the same lines as exploring ways to transplant patients with ADA SCID or reticular dysgenesis, or those types of SCID.
Okay.
And also, from a scientific standpoint, from the patients who had been previously transplanted, if they had some difficulties with it in grafting and being intact, this is also a great way to demonstrate that the use of 191, if there's better engraftment with our antibody, it's a good way to demonstrate that it is actually helping and working. So you can compare same to same, that if the first time that they received that donor engraftment, if it didn't engraft well, but then it does with our antibody, then it really does help demonstrate that our antibody is working, and thankfully that's been the case. But, as Wendy said, we're definitely going to be expanding and trying to make this more flexible as we continue going forward. Keep in mind, we're not approved yet. It's still an investigational product, so we are held to certain boundaries and restraints to make sure we do things in a very methodical way. Otherwise, the FDA will beat us up, and also it wouldn't be appropriate, so we got to do it the right way.
Well, the one thing we're all familiar with is we know what clinical trials are, so we get that part of it, and trying to get your FDA approval. So do you see the monoclonal antibody being used for other types of SCID, particularly ADA? So this is another ADA question, which you've kind of answered, but maybe you could just elaborate a little bit more again.
Yeah, so we are really actively exploring ways to combine other agents with our antibody, such that we could get engraftment in SCID patients with ADA, for example, and for patients with other diseases that could benefit from transplant as a whole. And so I think, that's up and coming, and we've been studying and we've published some exciting data about potential combinations, and we are working through, in our development program, how to basically fit that into SCID as well as other diseases.
And this is kind of along the same lines. So someone's asking, so would you retransplant with an autologous, so with the patient itself, instead of using an allo transplant with the original donor? So what if the original donor wasn't available?
Yeah, so I think we are actually also exploring, ways to bring our conditioning agent to gene therapy, and I think it would be very exciting, to bring this into gene therapy. But it's still, a work in progress, and we're discussing with some of the preeminent gene therapy for SCID, investigators in the country. Actually, some of them are on our scientific advisory board, Dr. Harry Malik at the NIH, for example, Dr. Mort Cowan at UCSF. So we are definitely exploring these opportunities with them and seeing where we can go. So I'm hopeful that we can move something forward.
Great. Will myeloid cells change to donor after retransplant using that antibody, and does that cause any issues or future issues for patients that had previous unconditioned transplants?
So the myeloid cells, they are not, I would say, specific to any particular virus or bacteria or any particular cell type. They are kind of, they're agnostic in terms of what they fight off. And these cells, so whether they're donor or host, functionally it doesn't matter, but we use it basically as a proxy for how well we got the healthy donor stem cells in, because those donor stem cells make all of the immune cells. They make the myeloid cells, but they also make the T cells and the B cells. And so we're looking for them to make the normal B and T cells in SCID patients. But we use the myeloid cells as a proxy.
Mm-hmm.
Okay.
I mean, Heather, if I could just, I mean, if you don't mind me just adding a little more to that, is that myeloid cells, it's not so much they're changing. You know, like on the previous slide, you know, the whole point of the antibody is to clear out all of those cells that make all of our blood cells, those hematopoietic stem cells. And the reason we focus so much on those myeloid cells also is that they're the ones that turn over very quickly. In other words, they have very short lives, and we really rely on our marrow. But they're not necessarily changing as much as being replaced. I don't know if that helped add a little more light to where we're coming from.
Okay. And for the person that asked the question, if you need it rephrased or anything, please send me another message, and I'll be happy to relay that. Anything that would just disqualify a patient from this trial?
So right now, if it's a retransplant subject, besides the genotypes that we already talked about, if that patient has significant immune function, so they have a lot of residual immune function, they're not, to the levels of what we would call severe combined immunodeficiency. And by that, I think the term is leaky. So then we evaluate on a case-by-case basis how the immune function is. You know, if there is a very high level of inflammation, or basically immune cells that are making a lot of signals and causing this activated immune system environment, we do think very seriously about, is our antibody alone conditioning going to be of benefit for that patient. So we do measure the immune cell function before transplant to see how, where the baseline is for that patient.
Okay. Are there any risks associated with receiving the antibody?
So at this time, I would say that the antibody has been, so well tolerated, and it's not what we call genotoxic. Basically, it's not damaging DNA or causing any of those kinds of issues. I think the risk is minimal to none, essentially.
So
I'm not saying minimal because I can't predict the future for everybody, but really for the 12 retransplant patients and the 2 newly diagnosed patients that we've dosed to date, there haven't been, any risks that arose from the
Yeah, I mean, Wendy, I think it's very smart that we choose our words carefully, because we don't, it's still an experimental procedure and it's in development, although we've been very fortunate, and the patients who have had access to it in the program have done quite well. It is important, though, to point out, this has actually been evaluated in healthy volunteers before Jasper took control of the antibody. It was being developed for something completely different that still had an immune process involved. And so it was evaluated at much higher doses. And thankfully, again, there had not been any evidence of any significant adverse events. And I think, one of the areas that we want to emphasize, I'm sure all of the families who are on the call now are always concerned about is, at least relative to some of the other conditioning regimens, like Wendy said, we have no reason to believe that it will cause any long-term side effects. We don't have any data to support that at this time. So it definitely appears to be a step in the right direction from a safety perspective and long-term risk. By the way, I don't know if everyone else saw this, but Jennifer asked a question about cord blood transplant recipients that popped up on my screen.
Oh, okay.
So I'm sorry. I don't. Are we allowed? Or did you want to handle that? Heather, did you see it? It popped up on my screen.
Yeah, I haven't gotten into it. I still have a slew of other.
Oh, good. All right, then keep going. I don't want to take you off target there. A lot of good questions coming up.
Okay, and I don't know if this got answered already or not, but I'm gonna ask the question exactly the way it's written, and then I might rephrase it a little bit too. But the way it's written is: has anyone received gene therapy post JSP 191 or only donor cell transplants? And then the way I'm rephrasing it is: if this did not work, could you receive gene therapy after this?
Well, the quick answer is, it's so far the antibody has only been used to condition for patients receiving donor, not gene therapy, the standard. But eventually,
and all of these patients, are still on our trial, so they haven't gone off to do, say, a gene therapy trial or anything like that.
Mm-hmm. But that is definitely something on our radar. I don't think we have any reason to believe it won't be a good conditioning agent for gene therapies. That's certainly something on our radar. And again, one of our scientific advisors, Harry Malik at the NIH in Maryland, he's certainly very interested in working on that as well with us. So there are options on the horizon.
Okay. Next question. Do you have data on the 4 reg patients? Which variant was the 20-year-old patient? Do you know?
So I don't have that information in front of me handy for which specific mutation in RAG1 that was. So I don't know off the top.
Would you be able to share that with me, Wendy, and I can share it with the person that's asked the question?
Yes, of course.
Okay. That would be great if you can. Let's see. Oh, and Susan's just telling me she's still muted, and that was 20 minutes ago. I'm sorry. I will let me ask one more question, and then I will get to it. Because I think she did want to add something, it looks like. Sorry about that. That's why I've got so many screens going at one time. Sorry, Susan. I did not do that intentionally. That was asked. Let's see. That one was asked. So cord blood transplant recipients are out at this point since the donor could not be available, correct?
Yes. Right now, they're not eligible for the retransplant group.
Okay. All right. Now you're saying, now I see
Robin
no
got
more.
the answer right. Robin got the answer right. I see that she responded correctly. For those of us keeping score at home.
All right. Now, what was the question, Kevin? You were saying
that was the one that popped up on my screen, and I saw the follow-up.
That was the
Robin WD, and
okay.
she got the answer right. But.
Okay. And Susan, you're unmuted, right?
I am.
Oh, okay. Okay. Did you want to say anything now that I'm?
Well, it's like back to the very early question. I just wanted, I was going to add just a little bit more context about the question about because the question was phrased, is it grown or is it manufactured? And the answer is both. And as Wendy said, it's grown in a cell. It's then secreted into the medium, the liquid, and then it's purified out of that. And then that is considered a whole manufacturing process where it's purified and then, made into a pharmaceutical grade, drug that can be administered. But so it's just kind of a complex. And then Kevin already addressed the live question. So anyway, like I said, it was going back a while, but I've been on mute. But so I just wanted to add that clarification.
Well, I appreciate that. And I'm going to show this last slide one more time, just in case anybody wants to see this again. And if anybody has any more questions, I can take them right now. Otherwise, I know that we've taken up a lot of your time, but I really appreciate you guys going through so thoroughly and in depth this presentation. It's a lot to absorb, but I appreciate the level that you presented it. Didn't seem too overwhelming, so that's great. And I hope that, oh my God, let's see, looks like, I think it was just a thank you coming through, not an actual question. So if nobody has any other questions, I don't know if Jasper, the group at Jasper, if you had any last minute thoughts that you wanted to say or anything, but we're so appreciative, like I said, of your time that you've taken to go over this with us. And, I wasn't sure what to expect. We've never done a town hall like this before, so this is our first one. Okay. So thank you for being that for us. And I think it's just kind of a neat way to get a lot of people with SCID and the different genotypes together at one time. And kind of the neat thing about this is usually you're talking about a subject where it would be just for SCID or it would be just for ADA SCID or something. So it's kind of neat to be able to have so many different of our groups be able to listen in and to learn something. So it is very informative and very appreciative. So thank you.
It's a real privilege to be able to work with this group, and I'm hopeful that we'll be able to make an impact. That's why we're doing it.
Yes.
Yeah, thank you so much for the opportunity to share our program. And, we are so thrilled and excited to, be able to, study this drug and, hopefully make a difference in these patients' lives.
Yeah. Thank you. Very exciting. So wish everyone a wonderful evening, and thank you again.